All right, we'll just give it one more second while everyone's connecting. I think we have one more person connecting. All right, great. So hi, everyone. Thanks for joining us today for our UK Cares webinar series. I'm going to introduce Dr. Alison Sanders, who's an assistant professor in the Department of Environmental Medicine and Public Health and the Department of Pediatrics at Icahn School of Medicine at Mount Sinai. I've actually met Alison earlier in the year. So Aaron Haynes put us in touch, and Alison gave a great career development type talk on networking. So we've already used her services earlier in the year, and then we get her to present here again for our webinar series. And just a quick reminder as I start to introduce her is I already see everybody's muted and your cameras are off, and that's great. And I'm going to go camera off too, just so we have the best connection. But please start putting questions into the chat box if you have them as we go through, and then we'll have some time for discussion at the end as well. So Dr. Sanders has an extensive training record. She got her bachelor's degree in biomedical engineering at UVA, so in Virginia. Then she went on and got a master's of science in civil and environmental engineering at University of Wisconsin-Madison. And then she sort of has a dream team of environmental health mentors and mentor background. So she got her PhD in environmental sciences and engineering in Chapel Hill at University of North Carolina under the supervision of Rebecca Fry, who many of us know. And then she went on to do a prestigious fellowship with Bob Wright, who many of us know, and he's given a seminar with our environmental health seminar series here, and then also with Dr. Lisa Satlin, who I don't know, who is in pediatrics at Mount Sinai. So she did that until 2017. Then she was awarded a K99 R00 for some of the work that she's going to talk to us about today, and then took on a lecturer role and then became an assistant professor in 2018. She has an extensive publication record already. So even though she's only a junior faculty member at Mount Sinai, she already has over 30 publications. I'm guessing it's close to 40 now, as I have a CV from back in February. And then I was complaining about having to submit this Victor grant because I was the submitting PI, and when Allison and I talked a few months ago, she told me that she had just submitted 4 R01s. So that kind of put it in perspective for me that maybe I should just stop complaining and start working a little bit harder. So I was impressed by that. And I know we're going to hear about her science today, but one of the things, me being the career development leader for our UK Cares, I love the career development aspects of her CV. And she's the founding director of their interdisciplinary environmental health postdoctoral fellowship professional development program. So she's working a lot with the postdocs to make sure they're prepared for their career. And I told her I was nervous to introduce her, and she said that's one of the topics for their postdocs. So I may attend that session next time. And she's also the associate director of their T32 fellowship program in pediatric environmental health. So she has lots of awards and things on her CV, and she's gotten some good scores on these R01s that she submitted. So hopefully in the next couple of months, she'll revise, resubmit, and then be super well funded, and then everyone will recruit her all over the country. So I hope my introduction was good enough, and welcome Dr. Sanders, and take it away. Thanks.
Thanks so much, Kevin. I appreciate that. And I actually meant to remind everyone that if you're a trainee PhD student thinking about postdocs, definitely reach out to me anytime. I'm always recruiting for my lab, and can connect you with others if my research isn't of interest to you. Actually, one of my current postdocs, Dr. Maria Politis, was formerly from the University of Kentucky. So glad to have more recruits anytime. So today I'm going to be talking about developmental exposures, toxicant exposures, kidney function, and biomarkers of cardiorenal signaling. I'll repeat some of Kevin's introduction about my journey through academia. So I'm from the DC area originally, just outside of, in the Virginia part of the suburbs of DC. Beautiful place, lots of monuments, also some, environmental health challenges there with things that we're going to talk about today. I'll talk about where I started my academic career in biomedical engineering at the University of Virginia, a much smaller town, so only 48,000. And as Kevin mentioned, here I was doing basic cardiovascular science, and I really I gained a lot from that experience, but really learned what I didn't want to be doing. So I was really interested in public health applications of my work. And so this is when I transitioned to UW-Madison, where I then focused more on environmental engineering. And so here I sort of created a hybrid program. I was doing a master's in environmental engineering, but also a global health certificate where I got to work with communities with infrastructure challenges specific to water in places like El Salvador and rural Wisconsin. So I worked with a number of Native American tribes in Wisconsin and Michigan. And this really led to my interest in how water quality, which I'll talk about today, impacts human health. It's also what led me to go on to do a PhD at UNC. And so here again, I sort of made a hybrid program where I was doing a PhD in environmental sciences and engineering in a school of public health, and I minored in epidemiology. And so the first study that I'll talk about today is how we looked, some data that we looked at to understand how well water quality, specifically toxic metals, impacts pregnant women and developing children. Next, I moved to New York, where I've been for 8 years now, a much, much larger city. I completed my training here and also began my faculty career. For the last 4 years, I've been directing a research lab for environmental toxicology. This work is supported by an R00 award, as well as a recent NIDDK R01, on which I'm a co-I, to examine environmental exposures in kidney health, especially among susceptible populations like preterm infants, children with obesity, and also racial ethnic minorities. I know that as a community of scientists, there's a lot of focus on neurodevelopmental, obesity, and respiratory effects of environmental toxicants. And from the nephrology community in the last recent years, we've seen an increasing appreciation in terms of long-term consequences of poor early life kidney health leading to later life disease like hypertension and chronic kidney diseases. And so my research is tackling this niche at the intersection of kidney development and the environment with the long-term goal of reducing the burden of chronic disease. And so that brings me here today to lovely Lexington, where the population is more in this mid-sized range. So I'm super excited to be here again for a second talk. I did get the opportunity to travel through a few months ago in between these two lectures. So it's been, one of these times we'll align it all so we can do the talk and the visit at the same time. But today I'm going to talk about sort of the overarching work that I've been conducting over the last 15 years or so. So my research broadly uses transdisciplinary approaches to study environmental metals and early life health outcomes. And so today I'm going to share 3 main areas. The first is exposure science, where we're looking at levels of arsenic and other toxic metals in private wells in North Carolina. And we'll examine the link with birth defects as some of my early work, and then work from my current laboratory that focuses on the impacts of toxic metals and other exposures on critical developmental windows of susceptibility that are particularly relevant to the kidney. And lastly, I'll talk about the third arm of my research, which focuses on novel kidney biomarkers. And so overall, I hope to show you the synergy of these areas in the developmental programming of kidney health and disease, which is the nexus of my career and the laboratory that I direct. So I'll start by talking about toxic metals generally. Hi, Erin. What are they and are we exposed? So toxic metals are metals that have no biological role and therefore they're not essential minerals. There's no beneficial dose for a toxic metal. And so I'm showing you here some examples, things like arsenic, cadmium, mercury, and lead. And I'll talk about each of these today. But what I want to note through this image of the periodic table of elements is that there are other toxic metals that appear on this table. And I'm highlighting beryllium in the upper left since it's an example of a light metal that is toxic. So sometimes we hear people talk about toxic metals using the word heavy, so heavy metals. But today I'm going to refer to them as toxic just to stay on the same page that you can be a light metal and also be toxic. So I'll talk about arsenic first, and then we'll get into some of the others. So arsenic is a metalloid, meaning that it has physical and chemical properties of both a metal and a non-metal. It's the 20th most abundant metalloid in the Earth's crust. It's also a known human carcinogen of multiple target organs, the bladder, lung, kidney, and skin. And it also is associated with non-cancer endpoints, so things like cardiovascular disease, diabetes, neurological abnormalities, and skin hyperpigmentation, which is the picture that I'm showing you here of one of the classic symptoms of arsenic poisoning is this hyperkeratosis on hands and feet. We also know that sources of arsenic are prevalent. So we've seen a lot in the news lately about arsenic in treated wood, which has been mostly removed from playgrounds in the US. There's also sources of arsenic that can include pesticides and herbicides, industrial processes like smelting and coal emissions. And in the last 6 months, we've seen reports on things like arsenic and other toxic metals in baby food, reports on juices, rice, seafood. And the source that I'm going to talk about today is drinking water. So my question to all of you is, which of these 2 glasses contains arsenic? And I always get lots of answers to this. Sometimes I hear both, sometimes I hear neither. And the answer is, generally we can't tell. It's probably both. Public distribution systems are regulated, but they have an allowable level of arsenic, so it's likely both. Whereas domestic wells are federally unregulated, which is what I'll talk about today. So here's a map of arsenic contaminated aquifers around the world. They're shown in peach color. A striking example of substantial endemic arsenic exposure has been in West Bengal and Bangladesh, where over 77 million people have been affected by arsenic contaminated water supplies. So from this map, if we look over at the US, we can also see that, yes, there's some areas in the Western US, but we also know that there's areas in the Eastern US that aren't highlighted. And that's because there have been numerous regional studies of arsenic in groundwater across the US that have shown that there's smaller pockets of naturally occurring arsenic, including in the study that I'm going to show you here that took place in North Carolina. So in North Carolina, we have about a quarter of the state population, or 2.3 million individuals, that use domestic wells for their drinking water sources. This means they're federally unregulated wells. And in Kentucky, just for comparison, there's about 70% of the state population that uses domestic wells, so federally unregulated for contaminants, and that's about 700,000 people. So North Carolina is the 4th largest state population that relies on private domestic wells for drinking water. And so we worked with the Department of Health and Human Services in North Carolina to comprehensively determine spatial and temporal trends of arsenic, but also other metals in North Carolina. And this collaboration started out because the DHHS for the state of North Carolina had a ton of data, over a decade's worth of data that they'd collected since the 90s, but they had no system in place to examine the data more closely and identify areas at risk. So we had over 68,000 previously measured well water samples that were analyzed with ICP-MS, and through a number of months and years, geocoding, assigning coordinates based on street, address, zip code, or county, we were able to map all of these. So that's, about 3 years of my life as a trainee produced this map. So we were able to produce one of the largest studies of arsenic in North Carolina to date, or the largest. And what I'm showing you here is the wells that measured above the EPA standard are shown in black, and wells that were at the EPA standard, which is 10 parts per billion or below, are shown in gray. Sometimes they're hard to see depending on everybody's computer, but you might notice there's a pattern to this banding of wells with arsenic down the state, with that very large spot down in the south central area of the state, and that's right around Charlotte. What we found was that over 12% of the wells in the database had detectable levels of arsenic. So if we think about that, giving the information that I shared earlier, which is that 2.3 million North Carolinians rely on private wells for drinking water, and 12% of these wells have detectable levels of arsenic, that's about 250,000 individuals who are at risk of consuming water with detectable levels of arsenic. We also found that over 1,500 of these wells exceeded the EPA standard of 10 micrograms per liter, or parts per billion. And similarly to lead, which has an EPA standard, there's no safe level of arsenic, and that's especially true for vulnerable populations like children and pregnant women. So in just this simple map, we were able to look at geographic trends across the state. Through the quantitative analysis, we found that there were levels as high as 800 micrograms per liter, which is about 80 times the EPA standard. So in a follow-up study, we also wanted to ask, what about other metals, and can we identify trends in other toxicants across the state? So here I'm showing you arsenic, cadmium, manganese, and lead. And if you look at panels A and C for arsenic and manganese, you might also notice a similar pattern. So these two metals, arsenic and manganese, coincide along a geographic area known as the Carolina terrain, which goes through the central area of the state and extends down into South Carolina and Georgia. The map of arsenic is similar to information as what we saw before, this banding pattern, which has higher concentrations closer to the south central areas of North Carolina. Manganese follows the same pattern. Cadmium can be introduced through groundwater through urbanization and anthropogenic activities. So it's also interesting that the highest levels occur near major cities. So you've got Charlotte down here and Raleigh up here. And then in the map of lead, lead in drinking water largely depends on the age and material of the pipe being used. So it's not surprising that these lead levels are more randomly spatially distributed. When we asked how many of these aggregated or each of these wells exceeded the EPA standard, we found again that there was about 2.4% of wells exceeded the EPA limit for arsenic, which is 10 parts per billion. A very small fraction exceeded the 5 parts per billion standard for cadmium. Around 21% exceeded the secondary maximum contaminant level for manganese. So many wells exceeding this 50 parts per billion standard for manganese, which is not a standard based on health effects, it's a standard based on odor and taste. So yet for manganese, there are no health-based guidelines for that metal.
Allison, I'm sorry, I've got a question.
Hello, yeah.
Hello. So you said a lot of the wells down there in the southeast, you said they're near Charlotte, but I'm assuming they don't drink from the wells. So is it that, they don't care about the wells because people are, drinking bottled water or faucet water instead? Or, I mean, how do you think about it?
Yeah, I mean, in a lot of places, that might not be an option to just drink bottled water. And in many cases, especially with arsenic, because it's odorless and tasteless, people can be drinking arsenic contaminated water without knowledge. So one of the primary outcomes of this study is that we really did a lot of PR to get information out there to communities that were most affected, and then also to encourage well water testing if that's available to you, but also to try to start working with government organizations to help provide reduced costs for that testing, because that can also be expensive, of $100 to $200 per water test.
Yeah, and Dave Orrin makes a nice point in the chat about how the one county right by Charlotte is very rural as well. So yeah, I mean, I think that this data is fascinating and scary all at the same time, because I remember that I tested our water at my house and they never sent the results back, and I was like, I wonder if they were so bad that they just didn't send them back, and whether there's screening that takes place by whoever's running the analyses.
Yeah, it's really interesting. I mean, sometimes it's similar to some of the test results you might get, at a clinical lab, that the only things that are flagged are if they're above a certain level. So if for example, the arsenic standard is set at 10, but you've got a level of 5, they might not send it back to say, hey, there's arsenic in your water. They just might send it back, everything's good here. So it gets into some really interesting questions about how these things are, actually not regulated and why they aren't regulated.
I like when people break up the talk, so I hope you're okay with us interrupting. We kind of like to do that. But I guess, so I don't really know this, and I probably should, so I'm going to ask you. So like if we're looking at fluorinated compounds, we might add all of them up, right, to come up with some kind of value. So are these metals all acting through similar type mechanisms or pathways that you can come up with some kind of additive value of all of them, or is that not really done or isn't important, I guess?
So I'll get to some of that when we start looking at mixtures in the next section, but
Okay.
no, we can't say that they're all working through the same pathways. Many of them work through different pathways in the body, but that hasn't prevented us from looking at metal mixtures from a more statistical and joint effects methodology.
Great, thanks.
Mm-hmm. So I'll just finish up saying that what we just had this conversation about, that we were really, yes, we published some papers, but what we also did was develop an online resource for homeowners. So people who live in these areas can now go online and find publicly available maps for not just the 4 that I showed you here, but over 20 different contaminants and information that's specific to their county. So I, as an early career scientist, learned the process of taking a database that wasn't explicitly designed for GIS, and through many years of stringent filtering, geocoding, and learning GIS tools, was able to map the data for public health surveillance. And I also learned the importance of working closely and collegially with a state health department. So that was the North Carolina DHHS. So without them, we wouldn't have had these data, and together we were able to craft more meaningful messaging. Yeah, is there a question?
Yeah, I was wondering, so on your website, do you also have like, so if somebody is drinking contaminated water, do you say you can use this water filter and get the lead out or whatever?
Yeah, so we also, that's another thing that we recommend is point of use devices. Again, if you can afford them, they range in cost of how fine you want to go with what you're looking to remove. Arsenic can be expensive to remove and might require a reverse osmosis filter, but other things like lead, Brita filters, all those other things you can buy at Target and Walmart are rated to remove lead, and some of them are rated to remove cadmium as well. Okay, so that was mostly about exposure, and so then, or environmental occurrence. And so now we're gonna move into, are humans exposed, which is what we really care about as public health scientists. So we conducted a subsequent study, also again with the North Carolina DHHS. My dissertation work was on birth defects. So some of these slides are gonna refer to birth defects as the outcome of interest for me. I was really interested in these metals as understudied developmental toxicants, which is now, been strung through my faculty career as well. So the pie chart on the left shows just a general breakdown of the known causes of birth defect, that 20% are due to genetic factors, 10% are due to environmental factors, and then this large piece of 70% that are due to unknown factors. And this is a real challenge in the area of birth defects, and we think that probably the appreciation for environmental factors is underappreciated. And we think this to be true because metals rank consistently in the top 10 of the priority list of hazardous substances from the ACSDR. Arsenic, lead, and mercury at the top, with cadmium down at number 7. And so we went ahead and did another study looking at are pregnant women exposed to these metals. So in a sample of just 211 women in North Carolina, we measured blood metal levels. And I'm not showing you all the data, but just that around 60% of the women had detectable levels of 1 or more metals. And notably, 100% of the women had detectable blood lead levels. And these data are pretty similar to what we see in nationwide studies, but we also wanted to have good evidence that this was also occurring in North Carolina, because we're seeing environmental occurrence, but needed to also show that there were exposures among susceptible populations like pregnant women. And this was a relatively small study, but it wound up helping to shape state screening policy for prenatal lead exposure in the state. And so pregnant women in North Carolina now complete a questionnaire and undergo prenatal blood lead testing if their responses indicate potential exposure. So even these kind of smaller studies that are just descriptive can actually wind up having public health impact in areas that are worthy of study. So I'll just end this section by talking about briefly where my dissertation headed. I had an interest in studying metals and associated birth defects. And that's because we know from a number of animal models and some limited prior research that metals are associated with or can cause gross morphological defects. And so here in the top right, I'm showing you a lab that we conducted in Dr. Fry's lab, in which we treated embryonic chicks with arsenic. And those chicks went on to develop anencephaly, a severe defect of the brain and skull. We also know from prior research that zebrafish treated with both arsenic or cadmium can induce heart defects. And so the white arrow there is pointing to an enlarged cardiac region. The red arrow is pointing to a curvature defect, a defect of the spine and cerebrospinal fluid, which is unique to zebrafish. And then when we overlaid these meta level data with an active statewide surveillance system of birth defects that had also collected data for over a decade, we found that there was indeed this really interesting putative relationship between areas with higher manganese in well water and a higher prevalence of a certain type of heart defect called conotruncal defects. And so this was an ecologic study. We were using aggregated data at a geographic unit, but it identified these potentially interesting and suggestive associations that I'd love to continue following up on, especially with relation to manganese and cardiac effects. So here I'm going to transition from some of this work on birth defects into more of my ongoing research. And so from that foundation in birth defects research, I became really interested in the idea of critical windows. So can toxicants during specific windows of development disrupt development in ways that have later life health effects? And that's really the focus of my lab now, which focuses on toxic metals during these critical windows and susceptibility of the developing and maturing kidney. And so that's important for kidney development because kidney development begins in utero around week 5 through processes of nephrogenesis and maturation. This continues to around week 36. At which point we're typically born with the full complement of nephrons we'll have throughout our lifetime unless damage occurs, and resulting in these two bean-shaped organs. There's then a number of years after birth where increased renal blood flow and eGFR happens sharply first through the first few years, continue to taper off until about age 20, at which eGFR peaks, and then continues to decline throughout the rest of our lives. And so within these windows of development that I'm describing, there's very discrete things going on. So nephrogenesis begins earlier on week 4 gestation. The majority of branching morphogenesis, so the early formation of the tubules, is completed during the 2nd trimester between weeks 11 and 14 of gestation. And then in the 3rd trimester is when 90% of nephrons, the functional unit of the kidneys, are formed. So that's happening typically between week 26 and 36 of gestation. And then of course, after birth, we have these increases in renal blood flow, GFR, and maturation that continues until around age 20. And so I'm interested in whether environmental insults during these susceptible periods, of which there are several, can program later life kidney disease or hypertension. And the NIH has said numerous times that how the fetal kidney environment contributes to the origins of CKD and adult disease is a critical research need. And I'm just updating it here to also include the perinatal environment. So after birth is really also important for the maturation of kidneys and healthy kidney function. And we also know that identification of early life risk factors like environmental factors are intervenable and therefore wield immense potential in both clinical and public health practice. And that's true because nearly 1 in 2 Americans has high blood pressure, according to the most recent guidelines. Worldwide, about 10 to 15% prevalence of chronic kidney disease. And then of course, end-stage renal disease is one of the most expensive outcomes to treat, either through dialysis or kidney transplantation. And I should mention that I'm not gonna talk about it much here today. I'd love to talk about it in the end or in questions, but a lot of this work is also interested, and I'm interested in health disparities research, since all of these hypertension, CKD, and even access to appropriate healthcare have evidence of health disparities by socioeconomic status as well as race ethnicities. So we know that metals are paradigm nephrotoxicants, things like arsenic, lead, cadmium, chromium, and lithium. are all prevalent environmental exposures. These metals occur concomitantly and occur from a variety of mixed sources. And maybe that gets to, Kevin, your question about, do they have similar mechanisms, which not necessarily, no, they don't, but they are still occurring at the same time in the general population. They're each can be proximal or glomerular toxicants. So those are different sections of the nephron, the functional units of the kidney. And they've all been associated with adult chronic cardiorenal diseases, but aren't well studied in younger age groups. In some prior work, we looked at combined exposure, so lead, cadmium, mercury, and arsenic in the NHANES population. So we're looking at adolescents aged 12 to 9. And we were interested in combined exposure. We looked at metals both in urine or blood. We had a total of almost 3,000 individuals in the study. And when we looked at the metal mixture on outcomes that included things like estimated glomerular filtration rate, blood urea nitrogen, and albumin, which can be sometimes non-specific indicators of poor kidney function, we found that there were these interesting associations between a metal mixture and increased eGFR among adolescents. This mixture was driven by mercury, cadmium, arsenic, and to a lesser extent, lead. But the major limitation of this study and a number of other studies in this field is that it's cross-sectional. So we really want to be able to look at exposures that predate the onset of any changes in kidney function. And I'll just mention here, because it came up earlier, that here we used a method called weighted quantile sum, or WQS, which is that index on the x-axis in the graph. And this is a mixtures modeling approach that accounts for correlation among the components of a mixture. And the ensemble approach addresses the combined effects of multiple exposures using this empirically weighted index. And so it gives us these really nice percents that we're able to kind of quantify how much each metal contributes to an effect. And we can also then parse out individual mixture components according to their relationship to these kidney parameters. And this is going to come up again because we continue to use similar methods. Is there a question in the chat?
So yeah, this is Dave Oren again. I'm really enjoying your talk. I just wondered, since you found that manganese often coincides with arsenic, are there any health effects known of co-exposure? And does any of your data address that?
Yeah, thanks. We did look at co-exposure in that initial study with birth defects, and we found that there was some evidence of interactive effects. But not many studies have looked at manganese and arsenic co-exposures, and it's super interesting in places like North Carolina, where you've got high levels of arsenic, you've got manganese that's not regulated based on health standards, but they are co-occurring. So great question, great idea. So moving on from the cross-sectional approach, again, I'm really interested in these metals and whether or not they can disrupt these developmental processes. So the development of nephrons, nephrogenesis, and then the primary processes that help us maintain nutrient and waste balance. So we want to examine exposures that predate the onset of subclinical kidney outcomes. And so one way that we thought to do this was to use the tooth biomarker, which some of you are probably familiar with since you've heard from, I think, Dr. Mindich in prior talks as well. And so this tooth biomarker uses a weekly resolution to give us metal levels starting in the second trimester when teeth begin forming. Teeth grow incrementally and form like growth rings on a tree. Circulating chemicals or things like metals are then deposited in these growth rings as they mineralize. And then teeth, while they grow, are continuously archiving what and how much and when an exposure occurred. So our goal in this study was to evaluate the relationship between perinatal toxic metal levels using this longitudinal tooth biomarker, so changes over time beginning in the second trimester, with children's kidney function. We looked at individual metals and then metal mixtures. And this study and some of the others I'll mention today takes place in the progress birth cohort. So this is a cohort in Mexico City, where pregnant women were enrolled during their second trimester, roughly 1,000 women. And we've been following the mom-baby pairs for over 10 years now. We currently have around 600 to 700 active mother-child pairs. And the data that I'm showing here are from a preliminary subset of children for which we had kidney outcome measures at the time of this analysis, and also available tooth data. So Dr. Christine Austin, together with Mindich Aurora, cut the teeth open and used laser ablation inductively coupled mass spectrometry to measure the metals across these growth layers in the tooth. And this allows us to reconstruct the history of metals exposure from the second trimester to about 1 year after birth at a roughly weekly resolution. Outcomes that we looked at, estimated glomerular filtration rate is a pretty standard one for kidney function, glomerular function specifically. We used a number of equations, but the one I'm showing here uses the biomarker serum cystatin C. We also measured serum creatinine, and we have those data too, but I'll just focus on one today. We then used reverse distributed lag models to look at these time-varying associations between weekly metal concentrations and children's eGFR, and adjusted for a set of standard and important covariates that we apply in these studies. To look at the time-varying metal mixtures, we then applied a newer method that Chris Jennings has published, which is the lagged weighted quantile sum regression. And this allows us to look at the changing mixture effects over time. And I'll show those results. So again, it's the progress cohort. The children are around age 9, predominantly low SES using standards from Mexico. Around 30% had exposure to some cigarette smoke in the home during the second trimester. The majority were considered normal weight, and nearly all of them had a relatively healthy, not clinically significant level of kidney function, so in good health. And so here I'm showing on the y-axis the effect of lead on eGFR, our primary outcome, and on the x-axis is time since birth in weeks, with birth denoted in with the red line. And what we found was that we identified in the 7 weeks prior to birth an association between prenatal lead exposure and increased eGFR. And it's super interesting to me, being interested in these critical windows, because this occurs during that third trimester period of nephrogenesis, so the formation of new nephrons. When we looked at some of the other metals individually, these lines are relatively flat, so not much going on with cadmium, arsenic, and chromium individually. With lithium, it's a little hard to see here, but towards the end of that timeline, postnatal lithium does seem to have an inverse relationship with eGFR, so higher lithium with postnatal reductions in eGFR. And then what was really striking was that when we looked at the metal mixture effects, so not just individual metals, but now combining them all together, we see that this metal mixture exposure that occurred in weeks 16 to 30 after birth was associated with decreased eGFR, which would be an indicator of reduced kidney function. And so this association was driven primarily by lead, which is the pink line there on the right-hand side. So this is again, the time varying weight in the mixture. So lead is highest in these weeks 16 through 30, or the greatest contributor to the association, followed by cadmium, chromium, lithium, and to a very little extent, arsenic. So a lot of that requires further assessment. So that was a single time point. We have not yet evaluated kidney function trajectories and whether those are predictive of later life disease, but that's a goal in this cohort, and any other cohort that's interested in collaborating. We're really interested in that early adolescent period when these trajectories can predict later life risk for CKD. The outcomes that I showed here are for glomerular endpoints, but we also have data on tubular and hope to collect data on vascular kidney damage indicators. And this work and some of the RO1s that are in submission are interested in whether there are potential unifying mechanisms. So they may operate through different modes of action, but are there unifying things like inflammation and oxidative stress, which we hear a lot about in the environmental realm anyways. and how does that affect developmental processes during these very specific windows. And so I'll mention that too. What I showed here was really focused on metals, but we've also looked at other early life environmental exposures. We did a study a few years ago with Dr. Ashley Mallon, who's a former postdoc, where she looked at both plasma and water levels of fluoride, again in NIEHS, and looked at kidney function, again in adolescents. And this was to date the largest cross-sectional study in North American adolescents. And what she found was a pretty striking increase per 1 micromole per liter increase in plasma fluoride. There was a 10 mil per minute decrement in EGFR. Again, these endpoints are not the most sensitive, so they're putative, especially in a cross-sectional study, and worthy of following up in prospective studies, which med student Charlie Saylor, who's currently in my lab, has been looking at this in the progress cohort. So now he's looking at urine fluoride assessed around age 4, so early childhood, predicting kidney function later in childhood. So some of the similar endpoints of EGFR, blood urea nitrogen, and blood pressure. And what he found for this manuscript that's currently in submission or revision is that there's no significant association between urine fluoride and EGFR, at least in this cohort and at this early stage. But he did notice that among children with obesity in a relatively small subset, that there was a marginally significant inverse relationship. So greater urinary fluoride with a 5 mil per minute decrement in EGFR. And so some differences between those two studies is that in the prior study that Dr. Mallon conducted, we were looking at plasma fluoride, which may better approximate bodily fluoride that's absorbed, as opposed to urine fluoride, which is more representative of bodily excretion. And also we know that younger children tend to excrete less fluoride. So the study that Dr. Mallon did was in adolescents. The study that Charlie's doing here is in pre-adolescents. And so young children excrete less fluoride than older children because more fluoride is absorbed in bone due to the rapidly growing skeleton system. We've also looked at links with air pollution. This is collaborations with Dr. Maria Jose Rosa, who's faculty at Mount Sinai, as well as Dr. Alan Just, also at Mount Sinai. And so here we're looking at critical windows of PM 2.5 exposure. There's been a lot of studies in the last few years showing that PM can cross the placenta and have effects on developing children. And so here we improved on some of the existing studies that had looked at discrete trimester specific findings to look again at this kind of smooth time during gestation. And so what we found with blood pressure as an outcome was that exposure to PM during these weeks in the 2nd to 3rd trimester of weeks 11 to 32 were associated with increased blood pressure in early childhood. And this equated to a constant increase of 10 micrograms per cubic meter PM 2.5 sustained throughout this window would mean a cumulative increase in about 2.5 millimeters mercury of systolic blood pressure. So really small changes, but small changes that have been shown when they start this early in life to predict subsequent trajectories for things like early life elevated blood pressure leading to adolescent hypertension and young adult hypertension. When we looked at some of the kidney outcomes, this is a study that's now under review as of Monday. We also found that there were some critical windows related to kidney function. So not just blood pressure, but the first weeks of gestation, so weeks 1 through 18 were associated with increased EGFR. And then when we look postnatally, so this is by weeks. On the y-axis is EGFR and the x-axis is gestational age, with 0 being conception and 40 being plus or minus birth. Now we're looking postnatally, so birth is on the left side, and we're looking at aggregated years and air pollution exposure. And we find that this PM exposure in months 1 through 14 after birth are associated with reduced EGFR. And this is really interesting because that's during that critical timing where we have rapid maturation of renal blood flow and EGFR in that first year of life. So sort of putting this all together, there's a lot going on with metals, there's a lot going on with air pollution. And so, we've got lots of ideas for future directions of, are there unifying mechanisms for many of these exposures, these time points, and can we better capture some biomarkers that might help us disentangle some of what's going on in these early life stages? I mentioned both oxidative stress and inflammation, and those are all, great options that we're hoping to explore in coming years. So for the last 10 minutes or less, I'm hoping we can touch briefly on some of the novel biomarkers work that my group has been doing. And this is in repeated calls by the NIDDK over the last 10 years that there's an urgent need for new biomarkers of kidney disease. And they've categorized things as cell-based, functional, proteomic, metabolomic, epigenetic, and RNA-based. And this was an opportunity for me as a nice extension of some of the methods I had applied during my postdoctoral work in molecular epidemiology, where we were looking at effects of or links between microRNA expression and shorter gestations among pregnant women predicting delivery time. And the goal with this work is to extend this into children's environmental health, especially applied to kidney function. So some of the prior work was looking at these links in pregnancy and gestation. And I'll just say that the major shift between my prior work and the work that I'm showing here now is moving from intracellular microRNAs, so what's going on in the cells, and here we were interested in the cervix, which is an organ really involved in labor and parturition, to extracellular, so particles not in cells that are free-floating in urine, which makes them a potential non-invasive biomarker. And I just want to remind or let you know what microRNAs are. They are short, single-stranded bits of RNA that are typically 20 to 24 nucleotides in length. They post-transcriptionally regulate, meaning that they typically result in gene silencing, but not all the time. And they can selectively bind and target hundreds of messenger RNA, regulating over 60% of genes in the human genome. And I'm particularly interested in extracellular urinary microRNAs because they're promising biomarkers for subclinical kidney damage because they reflect kidney signaling and histological changes at the molecular level. And they've been shown to enable early detection of things like chronic kidney disease and progression of acute kidney injury. And so these extracellular vesicles, as they're called, are a heterogeneous class of lipid-bound particles that are shed by any cell in the body, typically under normal physiological and pathological conditions. They play critical roles in intracellular communication. They carry various active biomolecules like messenger RNA, microRNA, and proteins to target cells, and where they can then act on those cells. There's two main subgroups. One are microvesicles that are less than 1 micron in diameter, and the other are exosomes, which are smaller, and they're 30 to 150 nanometers in diameter. And I'm particularly interested in exosomes because they contain the major fraction of microRNAs in urine. These exosomes travel in bodily fluids, blood, breast milk, as well as urine. So they're a great potential target for molecular biomarkers of kidney disease. We know that exosomes in urine are primarily released from renal epithelial cells that are coming from tubular structures in the nephron. One caveat is that they can also be coming from the urinary tract, so it's a little difficult to disentangle now without more sophisticated methods. But they're one component of this idea of a non-invasive liquid biopsy, so a simple urine sample being able to tell us something about what's going on for intrarenal signaling. The exosomes in urine contain microRNA from the glomerulus and all sections of the nephron, and we can measure over 750 in a single assay. So we don't know exactly what these microRNAs are doing in the kidney yet, but there's been a number of studies, both in vitro and in vivo, that have shown that microRNAs encapsulated in exosomes can mediate blood pressure, as well as sodium, potassium, and other ion channels in the nephron. So we do believe that they are functional. We just don't know exactly how yet. So our study goal here was to look at the relationships between urinary exosomal microRNAs with cardiorenal parameters in kids. We're again in the progress cohort, but now it's a subset of 105, just because of the cost and also the time involved in these types of analyses. So this is Dan Flores, lab manager for my group. He had the privilege of spending an entire summer isolating urine exosomes. So we start with 10 mils of urine and wind up with a very, very small amount of 12 microliters. So this is a very laborious process for him. It's one of the most rigorous methods that can be used. That's differential centrifugation to isolate exosomes. it's still considered the gold standard, although a lot of labs have moved to a simpler method where they're using filtration steps. But we are hopeful that this method is the best that we can do at this time, and also gives us much cleaner exosome isolation, whereas some of the other preps can lead to kind of a more heterogeneous mixture of larger extracellular vesicles mixed with exosomes. So this method is really good for specifically exosomes. They're really tiny particles that are 30 to 150 nanometer enrichment. So we do a couple cleanup steps, and we measured them on open array qPCR. We also visualize them using transmission electron microscopy, which I'll show you an image of here. So we found indeed that we could image the exosomes from this long summer that Dan spent isolating all these urine samples. And we found that indeed they match what we were hoping to see, that the exosomes, when we looked at the size distribution, matched sort of the distribution we were expecting. So this is good news for Dan, that the method worked, that his summer wasn't for naught. We then went ahead and measured them using qPCR. We did some pretty stringent filtering and smoothing for statistical analysis, which I'm not going to go into the details of. But what we found was we detected over 150 extracellular microRNAs in these 10 ml urine samples from healthy kids. And when we looked at the microRNAs that were detected the most, we found a number of them were in the miR30 family, which is also great news for Dan and me, because they're the most abundant. miR30 is the most abundant microRNA in human urine. So that's also good that the protocol worked. We identified what we thought we would. We had about 29 microRNAs that were detected in all of the urine samples, so 100% detection. And next we wanted to ask, what did these look like in relationship to some of the cross-sectional cardiorenal parameters that we had in the cohort? So we were looking at similar outcomes that I mentioned before, now including things like spot urine sodium and potassium. So we're really interested in whether microRNAs might be linked to ion transport in the distal nephron. And we also included the sodium to potassium ratio, which is sometimes a better indicator of cardiovascular risk than either sodium or potassium alone. We included blood pressure as well as eGFR. Again, we adjusted for covariates, and we applied some stringent thresholds to make sure that we weren't being overly ambitious with our detection rates here. We then asked what some of these microRNAs and their target genes do in the cell, and I'll just tell you briefly what we found. I'm going to just show the results for the exosomes that passed FDR correction. So this is work by Dr. Yuri Levin-Schwartz, also a postdoc working with my group, hoping to move on to new things quite soon. And what he found was that there were 3 microRNAs, exosomal microRNAs, associated with urine sodium. So a doubling of expression of each of these was associated with an increase in urine sodium. Notably, we found that there were 17 microRNAs that were associated with the sodium-potassium ratio. Again, interesting, because that's an indicator, potentially better indicator of risk for cardiovascular disease than either urine sodium or urine potassium alone. We didn't find any microRNAs or exomirs that passed FGR correction with systolic or diastolic blood pressure, but we did find one, miR27A, that had a significant p-value, just didn't pass our FDR threshold. And that was interesting because this microRNA has been associated with things like essential and pulmonary hypertension in adults, and in mouse models is related to increased sodium transport activity in distal nephron cells. And lastly, when we looked at the relationship with childhood eGFR, now we're doing a prediction. So 4-year exosome predicting later eGFR, we found one microRNA, miR520E. For each doubling of this microRNA, there was an associated decrease in eGFR of about 12 mils per minute. And that was interesting too, because although it's still putative, this family of microRNAs, miR520E, have been shown to be increased in individuals with preeclampsia, which is the pathophysiology of preeclampsia is characterized by glomerular dysfunction, which is we would expect to be associated with eGFR, reduced eGFR. So then lastly, when we asked what these genes do, what these microRNAs do, we found that a number of them are involved in disrupted ion transport and endothelial cell injury and inflammation. We identified a number of nephrotoxic pathways by which they might be related. So things like renal necrosis, renal proliferation, glomerular injury. Some of these networks included things like cell death in kidney cells. And notably, a number of the messenger RNA targets involved of these microRNAs are involved in insulin and aldosterone receptor signaling pathways. So again, there's lots to follow up on here, lots of putative things discovered in this relatively small exploratory study, but encouraging for future applications where we might consider looking at replication in cohorts with more clinically relevant populations, so children with CKD. This is the first study to look at exosomal microRNAs among healthy children. So, we're not necessarily looking at clinical outcomes. We're just showing that there are these really intriguing linear relationships. So interesting for potential future predictive novel biomarker work. And yeah, good news for the lab for proof of concept. So I'll stop there. I want to just come back to sort of the overarching public health implications of this work, which is that children with poor kidney health are at higher risk for failure to thrive, hypertension, kidney disease, and cardiovascular disease in adulthood. We're really interested in my lab in identifying these critical windows of development that can alter lifelong health trajectories, and also in the usefulness of identifying new biomarkers to help predict subsequent kidney health or nephrotoxicant exposure. So I'm happy to take questions, really interested in some of the directions this research will go in the next few years, and areas to extend this work into other cohorts. Can always write some more R01s. It seems to be a hobby of mine lately. And I'm really lucky to work with such a great team of collaborators at Mount Sinai. Some of the senior faculty I'll highlight here are Manish Arora, Chris Jennings, Lisa Satlin, and Bob Wright, as well as Mara Tejas Rojo, who's the other Progress PI in Mexico City. Marcela Tamayo Ortiz is my primary partner down in Mexico City, who helped get this kidney project started, as well as the members of the lab there at the Institute of Perinatology, who helped collect all these samples and prep them for a ship to New York, and Andrea Baccarelli, who's at Columbia University. Lots of funders, and very thankful for your support, your attention, and happy to take any questions.
So Allison, that was really awesome. And I'll just remind everyone that we are at 1 o'clock, but that's because several of us interrupted on the way through and had some discussion. So if you do need to hop off, go ahead. I'm sure Allison will welcome an email if you have questions. I see C2AO has a question, but he's already asked a few. So if anybody else has one before you hop off, go for it. And if not, I can ask C2AO. Here we got Dave. Go ahead.
So I already asked a few questions too, but I have to hop off in a second. I wanted to ask one other question. Great seminar. In your progress cohort, 30% of people, children, are prenatally exposed to cigarette smoke. Could you detect increased levels of cadmium, nickel, chromium, arsenic in those exposed children and their teeth?
We have looked at that, and so far, I think we didn't find that replicating, and I can't, we've had some discussion about why that might be the case. We know that the placental barrier is restrictive to cadmium transport, so perhaps that's one of the reasons why it's not tracking like we would expect it to. I think if I'm recalling correctly, we actually see higher levels of cadmium postnatally, and so we're trying to understand, the source of that, which seems to be different from the prenatal smoke exposure. But thanks for that question.
Well, thank you.
Mm-hmm.
Did anyone else have any questions? Because C2AO's first one was actually one I was thinking about, is whether or not the RNA biomarkers can be found in mouse models. So if you've looked to see if those replicate in any of the mouse models, so then you can have ways to do interventions potentially. Yeah,
that's great. It's maybe the direction one of our victors was headed. I'm super interested in not necessarily mouse models, but moving this into zebrafish models, just for simplicity, and also working with some collaborators to look at organoid models. So some of these 3D models of human-based cells. Yes, they do. They are present in mouse models and other animal models. So super interested in.
Yeah, or Ryan Semmel here at UK is also interested in microRNAs and things, but he has non-human primate models where he's looking at high-fat diet feeding and probably has some kidney damage and stuff. So he may be somebody you want to look into potentially getting samples from at some point.
Absolutely, yeah. Thanks for that suggestion.
Then C2A's other question was, is there a reason why you're not particularly interested in microalbuminuria as a marker of kidney damage?
No, it's a great indicator. It's more useful among individuals that have CKD. So it's there, when there are already glomerular issues, we know that's going to be present. But on healthy children, we haven't yet seen major changes in that as an outcome.
Okay, I guess I have one more question, and then we'll just make sure nobody else has any. But I guess I was thinking the whole time about low birth weight and IUGR and nutrition, I guess, and that was one of the things you didn't really talk about nutrition or calorie intake or anything during pregnancy. And I would assume if you modeled that somehow, that would have a pretty strong indicator of kidney health. So have you thought about ways that you can look at whether they're additive or whether metal toxicity or something is something that's causing some of the negative effects that are associated with IUGR or low birth weight or preterm birth or whatever?
Yeah, we've done a couple studies with low birth weight and preterm birth, not IUGR. At least in this cohort, there wasn't a high prevalence of IUGR, but we showed that for kids who had shorter gestations and were born lower birth weight, there was an interactive effect with lead exposure on predicting subsequent increases in systolic blood pressure. It's a really, you're right, there's going to be probably, I would assume, stronger effect sizes for kids who have had IUGR or low birth weight. Just in this cohort, there was a relatively low percentage.
That's kind of surprising, actually, with I guess what I would have considered an unhealthy environment in Mexico, because we have such high rates here in the US, especially in the South.
Yeah, I think that's been one of the surprising things of the progress cohort is that we have.
And I laugh because I don't want to cry, I mean.
We've not seen relationships between the primary exposure lead and preterm birth in that cohort, but that's we do see that in many other populations who are exposed.
All right, any other burning questions? If not, yes, I see C2A is giving a clappy hands, and I agree. I thought that was great. There's a lot of area for collaboration. I'm sure Ellen and Aaron are both sad that they missed the end of the talk, but you're doing some really great stuff. Let us know how we can continue to interact and engage. Sound good?
Yeah, thanks you guys for staying on the line. I appreciate it. Enjoy your Wednesdays.
Thank you. Have a good one.